Yeast cells can be genetically modified to produce a painkiller. This painkiller is usually obtained from opium poppies. One method for genetically modifying a yeast cell uses a plasmid containing the desired gene. Explain how a gene can be inserted into a plasmid. So basically a plasmid is a small loop of DNA you find in bacterial cells, and because it's small and manageable, we can easily manipulate it for the purposes of genetic engineering. So let's say we've got a piece of DNA that we're interested in, or a gene that we're interested in, because it contains the information to make the painkiller. So what we do is we'd use restriction enzymes. Those are enzymes which cut DNA at specific points. Remember, they are specific, so they'll find a specific region on the DNA, and they'll cut out the desired gene. And use the same restriction enzymes to cut out or make a similar snip on a plasmid. And what will happen is you'll leave, if you can see here, you leave a little overhanging region here called sticky ends. So there's something to bond with the complementary sticky end on the plasmid. So the DNA is inserted there, and then enzymes called ligase enzymes stick or bond them together to make one coherent loop of DNA. And then the bacteria multiplies and multiplies, because that's what bacteria do very quickly. And with them, the gene multiplies, and therefore you can produce high volumes of the painkiller or the protein that is the painkiller very quickly. So how do you express this in terms of an answer? You say cut plasmid with restriction enzymes, insert gene into plasmid using ligases or ligase enzymes. Gene and plasmid must have complementary sticky ends. So today this is a 2-mark question, but this could easily be a larger question, or it could even be a 6-marker if they ask you a little bit more about it, maybe something about genes or about the benefits of genetic engineering. But here they've asked the benefits as a separate question. Discuss the possible benefits and risks of producing painkillers from genetically modified yeast cells rather than extracting the painkillers from poppies. So yeast cells are small and you can grow them in a lab very quickly. Poppies take much longer to grow, but with that there are issues as well. So greater yield as yeast grows very quickly. They're not weather dependent, so poppies are weather dependent, obviously, because they're flowers, they grow outside. They're cheaper because we're just growing them in a petri dish. They don't need soil and fertilizer, they just need a bit of nutrient broth, and they take up less space. So lots of advantages. So what could the possible disadvantages be? Well, painkillers may not be as effective if we're making them from yeast rather than naturally growing them, from poppies where they're normally grown. And GMOs could enter the environment, causing concern. Yeast are hard to keep track of, they're microorganisms, so possibly they could enter the environment. But these are more obscure and harder to think of. So just be aware of the kind of general argument here that if they're not grown in the natural way, it could be that they're less effective. And there's always concern with GMOs that they could enter environments and cause concern, but it's always a bit vague because we don't know the effect they could have. Here we have a structure of a DNA nucleotide. So this part makes up the sugar phosphate backbone, the sugar and the phosphate, and this is the base here, adenine, thymine, cytosine, guanine. It could be any one of those 4. Structure A is and that's a sugar. So this is the sugar, this is the phosphate, and this is the base. In 2003, the first complete human genome was sequenced. That means we mapped out the position of every gene that makes a human. The genomes of different people have small changes in the sequence of the DNA bases. These are called mutations. Describe how these changes in DNA sequence can affect the individuals, and how sequencing a person's genome can influence their medical treatments. So how can sequencing a person's genome influence their medical treatments? So this is a multi-layered question. We have to deal with this part first, then talk about this part here. So describe how these changes in DNA sequence can affect individuals. So change in a DNA sequence is a mutation. So let's start there. DNA has 4 different bases. Mutations are changes in the DNA sequence, resulting in different alleles for these genes. So different alleles basically explains why people look different, why they have different variation. DNA sequencing allows you to identify genetic diseases. So now we're looking at how it helps medical treatments. It helps you identify genetic diseases, identify the risk of developing disease, and can modify lifestyle. With this information, we can modify our lifestyle, reduce risk. These are just arguments you need to remember linked with the Human Genome Project. HGP impacts on medical treatment. So this is how it benefits us, or how DNA sequence impacts us directly. But in terms of medical treatment, HGP has determined the location of genes, so we now know where genes are. We have better understanding of some diseases, and we can provide tailor-made medical treatments for people because we know their genetic makeup, and basically the diseases they're more susceptible to developing because of their genes. So most of the marks come from actually just understanding the positives of the HGP, the Human Genome Project, and the benefits of DNA sequencing, and obviously just understanding what a mutation is.